My dear students, today we are discussing about one of the topic in genetics. So topic is the genetic testing. So what is the main aim of the genetic testing? So it is the identification of abnormal characters of genes, abnormal characters of chromosomes, abnormal characters of protein. That's purpose will go with the genetic test. That is the main aim. Just check the definition related to the genetic testing. Genetic testing is a type of medical test that identifies the change in chromosomes, genes, and proteins. Why you go with this test? Why go with the genetic testing? Identification of any changes, normal characters or abnormal characters, whatever it may be. Identification in the chromosomes, genes, or proteins. This result is genetic test confirm or rule out a specific genetic condition or help to determine person's chance to developing passing of genetic disorders, simply to estimate the genetic disorders. Genetic disorders. So lot of genetic disorders are there, like a trisomy 21, trisomy 18, Down syndrome, Edward syndrome, Klinefelter syndromes. Different types of genetical diseases and disorders is occur. That one is the in our body. Some genes are suspected genes is present. Some genes are act as a carrier. Some genes are exposed. So here every character is identified by the genetic testing. So total how many genetic tests are there? Approximately more than 1,000 genetic tests are currently available, and some are underdeveloped. Right. So what are the genetic tests? Genetic tests are mainly three types. So lot of genetical tests are there, but we'll go with the three are mentioned. One is the follow the genetic tests are 1,000 plus genetic tests are there, but every genetic test is related to the first of all basic structure. Basically they identify the which one. One is the molecular genetics, chromosomal genetics, and biochemical genetics. Total three different types of genetics are genetical tests are available. So first one is the molecular genetics. What is the role of the molecular genetics? Simply, what is the structural or molecular formula in genetics, that is called as the genes. This one is simply to study about the study about the to study about the single gene, means what is the length, what is the DNA structure, and what are their characters. So everything is identified the any characters in the DNA, any characters in the gene. So any mutations occurred, and which type of diseases and disorders is occurred, every character is determined. That is the molecular gene test. So only genes and DNA structure is identified by the molecular genetic test. Now we'll look at the chromosomal genetics test. Already mentioned chromosomal means to study about the chromosomes. What is the length? What is the DNA? See if there large genetic changes such as extra copy of chromosome is present, or one chromosome is less, or addition, deletion. This type of characters is studied under the chromosomal genetics. And biochemical genetics means to study about the amount or activity level of the proteins, amount and activity level of the proteins. So abnormal characters of the proteins in every character, simply to study about the molecular genetic test only study about the genes. Genes. Chromosomal only chromosomes. Biochemicals proteins. These are the main three basics of basic identification of the genetical problems under test. So why go with this test? This test is go with the which purpose? Reasons. What are the reasons? So first of all, finding the genetic disease in unborn child. See, at the duration of the pregnancy, fetus development, any genetical problems are there, just go with that purpose, identification. And finding out people carries the genes. Somebody already mentioned one word. Some genes are always not exposed to the environment. Some genes are exposed. So whose contains the not exposed genes, but they are present in the body, carry forward to the next generation. That one genes also identified. And there is one screening embryos for disease at the development of the at the duration of the pregnancy, at the development of the fetus. So embryo is perfect or not, that type. And the next one, testing genetic test adult before they cause the symptoms. So it one is a pre delivery, of post delivery also. Post delivery also, identification of any genetical problems, go with that. So testing genetic disease adults before they cause the symptoms, and making diagnosis persons who has disease symptoms, and figuring the type of dose medicines are best for suggestions. Medicines is suggested to the patients also. That's purpose will go with this. Simply priorly identification of the genetic diseases. Before birth, identification of the genetic diseases. Before the symptoms, identification of the genetic problems. Why? In case any problems are there, we rectify that type of problems with the help of the medications or any other methods. That's purpose will go with the genetic testing, like a different triple marker test at the duration of the pregnancy, TIFA scanning, and these are the scanning techniques are also determination of any abnormal organs is developed, the baby's fetus is perfect or not, and well developed or not, everything is identified. In case any abnormalities are there, at the time we'll go with the genetic testing, right? These are the some reasons related to the genetic testing. So what are the genetic types of the genetic tests? So 1,000+ genetic tests are there. Today we will discuss about few genetic tests. First one is a newborn screening. Newborn screening means after delivery, immediately to identification of any problems are there, just check. So means newborn screening used to the just after birth to identify genetic disorders that can be treated early in life. For example, phenylketonuria. Phenylketonuria. The phenylketonuria, it one is developed due to the deficiency of hydroxylase enzyme. Due to the deficiency of hydroxylase enzyme. This hydroxylase enzyme deficiency, it leads to the phenylketonuria. Phenylketonuria is develops. So this one is a inborn error. Actually, it is the inborn error. So this one type of problems, and then hypothyroidism related problems also determined after delivery by the genetic testing. This is the one. And diagnostic testing means the diagnostic testing used to the identification or rule out of a specific gene chromosomal conditions. So means here genetic test identification of genes, any gene abnormalities, chromosomal abnormalities are can be identified before the birth. This one is the before the birth, this one is the after the birth, and as well as the time of the duration of the person's life also identify the abnormal character. That is the diagnostic testing. And now we'll go with the carrier testing. Carrier testing is used to the identification of the abnormal genes, means mutation genes or carrier genes, one copy of gene, single mutation, or what, two copies of gene and multiple mutations are occurred, any genetical disorders occurred, and collect the family history, depends upon the family history also. So this type of genes is present in the body, or multiple genes or single gene, or which type of gene, everything is identified in the carrier testing. And one more is the prenatal testing. Prenatal testing is the testing detect the changes in the fetus genes, not mother genes, fetus genes or chromosomes before the birth. This type of test offered during the pregnancy, there is an increased risk that the baby will have genetic or chromosomal disorder. So these are the few genetical tests are there. So just go with the newborn screening, diagnostic testing, carrier testing, prenatal testing. These are the few example for you, and for the forensic purpose also, any criminal cases at the time also genetic tests will play a key role in the body. Identification of unknown sample DNA molecule, RNA molecule, protein molecules are estimated. So these are the few example for you. So this is about our today topic, and tomorrow we'll go with the one more topic. Thank you.